Take a little look at some of the metabolism that is occurring within the cells themselves within the body, and we'll take a little deeper look at some of the specific processes for ATP in another talk. But today we'll kind of just look at the general processes that are occurring within the body and kind of in the cells as well. So some of the things that we have to kind of build upon to start off with is the idea of energy itself. And so energy itself comes in two uh, main varieties that we're going to look at in terms of the body. And so we have kinetic energy and we have potential energy. And so kinetic energy is energy when things are actually moving. And so things are already on the way. If we can have both, in a sense, a potential energy and a kinetic energy resulting from it, uh, where we have potential energy stored away in things. And so this is where we have it in chemical bonds. This is where we have it in an electrical battery. So it's stored there. It has that potential energy. But once we start to use that energy, it can become kinetic energy. So kinetic energy can be an electrical energy as well in terms of electricity, and that movement of the ions that they are moving down their gradient there. Stored energy is kind of like taking water and pumping it up to a water tower, and we had to put some energy into it to get that energy there. Same thing goes with chemical bonds. We have to make the bonds themselves and impart some energy into it in order to get that energy stored inside there. But once we have all that water up at that water tower, now we have this potential energy. We have this stored energy in there, in that we can now have a large amount of water pressure. And so we don't have to keep a continuous pump running, in a sense, when everybody wants to turn on their taps. So we've already got the water pumped up there and it's ready there, giving us that potential energy. And the same thing goes when we have chemical bonds within the body as well. So when we make these chemical reactions occur, we have essentially two different varieties that can occur. And so we can have catabolic or anabolic types of reactions. For catabolic reactions are going to involve breaking that uh, energy that's been stored away. And so we have potential energy that is stored there. It's within the bonds themselves. And when we break those bonds, we're going to release all of that kinetic energy that was in there. And so that's going to give off a potential energy that's there, this kinetic energy that is occurring from it, from that energy that was there. And so this is going to allow us to do things like move things. And so we need this type of energy in terms of catabolic energy in order to move. So we need to be able to break bonds in order to cause muscles to contract. We need it for growth in order to build things up. We need it for reproduction. All kinds of processes within the body require us to break those bonds. But at the same time, we also need to be able to build bonds. We need to be able to get the bonds to be formed in the first place. And this part here requires energy. So it's requiring of energy. We have to get energy into the system before we can actually even have anything occur. So we're going to have to utilize that energy that's there in order to impart it into those chemical bond making itself. And so we are forming new bonds that weren't there before. And as we form those new bonds, we can then, in a sense, go from, now we have formed bonds, we can go from anabolic reactions to catabolic reactions. And so we can have that situation where we now have energy stored away, and now we can do those catabolic reactions to break them down and pull them apart. So the idea of catabolic and anabolic types of reactions is similar to kind of the images here. So if we were having sugar, and so we had sugars in a process where we're going to want to uh, make the sugars or break down the sugars. And so we're going to either synthesize the sugars, so something like a plant is synthesizing sugars, or ourselves starting to break sugars down to utilize their energy that's there. We're going to have two kind of different situations. And so the building up versus the breaking down leads us to a situation where we have either a synthetic process, and so synthesis where we are are making those bonds, those anabolic types of reactions. And then we have breakdown. We have those anabolic types of reactions occurring within the system itself there as well. So anabolic, building, catabolic, breaking down. So we're breaking down those bonds after we have taken them together. And so anabolic is always going to require us to put energy into the system. Catabolic is always going to give us energy back out. So we're going to take the energy that was in the bonds to begin with. And so plants are always going through a process of where they are making sugars. And so since in order to make something, we need to have some sort of energy added into the system, here we have solar energy. And so we have sunlight. And so that sunlight is going to take carbon dioxide. We're going to add water to it. We put the energy from the sun together, and we come out with sugar and oxygen. And so in the process there, we're developing an anabolic type of a reaction. We now have that plant there, and now other organisms like ourselves can take those sugars, combine it now back with the oxygen, and we're going to do an oxidation reaction here. And with that, we then have the ability to liberate the energy that was stored in the bonds. And now we get that energy out. We have water left over and then carbon dioxide. And so nothing is, in a sense, destroyed. Nothing is truly made. We're just simply changing where the process is. We're either doing a catabolic reaction or an anabolic reaction, and it's going around and around and around as we move essentially these bonds from one place to the next. We either build those bonds up or break those bonds down. So within that, we have the concept of endergonic reactions and exergonic reactions. And so endergonic reactions require energy. So energy is required into the system itself with an endergonic type of reaction. With an exergonic reaction, we're going to get energy released. And so we're going to have the release of energy for an exergonic type of reaction. This can be in the form of a exergonic reaction. Things like the temperature may go up, so things may heat up as we break things down. Endergonic reactions are going to absorb that energy. They're going to absorb any kind of heat into the reaction itself. In addition to endergonic, exergonic types of reactions and our catabolic and anabolic reactions, we can also have exchange reactions. In exchange reactions, here we're not really taking and breaking bonds per se. We're just kind of moving things around. And so the molecules that are within the reaction are just simply kind of rearranged. And so in the example here, we have AB plus CD, and we're going to take the D and move it over to the A, and we're going to take the B and move it over to the C. And so then we end up with our AD and our CB as our reaction, as our products for the reaction itself. This here can be either a catabolic or an anabolic reaction. So we can have decomposition and synthesis types of reactions occurring within the processes themselves. And so in this case, we're just kind of moving things around. We're not really generating energy in the process. We're not really breaking things down per se. This happens in the body at times when we have kind of excess amounts of energy. And so one of the things where we have this is ATP is a relatively high energy molecule. It's, in a sense, doesn't like to be that way. And so it's going to want to break down into ADP and inorganic phosphate. And when it does so, we want to be able to save that energy from that inorganic phosphate group. And so that phosphate group, we don't just want the energy to be lost in the process. So instead of just allowing that phosphate group to go on its own, we're going to add it to something like creatine. And so creatine is going to be there, and we're going to add that together. And so we end up with ADP plus creatine phosphate. And so that's our product. And then these reactions can then go back in the opposite direction. And so now we can take those products and move back in the other direction. Now when we have a situation where we need to have that energy from that creatine phosphate, we can bring that phosphate group back all the way over to the ADP and put it back on the ATP. So here we didn't lose any of the energy that was stored there initially. All of the energy was maintained within the system itself. And that allows us to just kind of shuttle energy around at times. And so we're just kind of transferring it from one place to the next. When we do want to be able to have a reaction go completely in a sense, one of the things that we may need to do is kind of push the reaction along. And so we may need to kind of help that reaction get started, so to speak. And so enzymes, a protein, are going to help to promote chemical reactions. And so they do this by reducing the activation energy. And so what the activation energy is essentially the amount of energy that we have to put into the system in order to get the chemical reaction to occur. And when it comes to enzymes and kind of the catalyst kind of a idea, one of the things with enzymes to kind of think about is if we wanted to have spaghetti. And so if we wanted to have spaghetti, we get some noodles, and normally you're going to take those noodles and put them in a pot of boiling water and boil them along. But in reality, we could get the same result just by sticking those noodles in cold water. The only difference there is that it's going to take a lot longer for the reaction, the water, to move into the spaghetti over time. And so it's going to take more time with it. And so the energy that we imparted by boiling the water essentially was high when we just put it in cold water. But although our heat isn't really an enzyme, it lowered the activation energy. And so the activation energy over here is a lot lower. And that's what an enzyme does, is it lowers the amount of energy that it takes to kind of get the reaction going, so to speak. And with that being said, we have to kind of think about our own body. And so in terms of our own body, we're at a relatively low temperature for a lot of chemical reactions to occur. If you're in chemistry lab, a lot of times when you're doing reactions and chemical processes, you're doing it with heat. You're adding heat to the system itself. And in order to get that to occur, it's a relatively large amount of heat to happen there. But we don't want that in the body. We don't want to be at 212 degrees at boiling, something like that. So one of the things that the enzyme does is it allows the reactions to proceed under normal body temperature conditions. And so we get those activation energies to start to decline, and we're then able to do those reactions inside our body itself. So the catalysts themselves, the enzymes, accelerate the chemical reaction. But the thing with the enzyme is not consumed. And so the enzyme itself is not being permanently changed or consumed. The enzyme that goes into the reaction is still going to be there in the end. And so until we get to a point where we have a stable product, that enzyme is going to be there, and it's going to help along the reaction itself. But in the end, that enzyme is still there, in a sense, unscathed in the process. So enzymes themselves are very specific to the reaction that's going to occur. And so when that occurs, the enzyme has a specific binding site for the substrates that are going to come in. So essentially, everything has to kind of look exactly right, which means that the binding site is kind of like, and the substrate are kind of like a lock and key. And so we have this situation where we have a specific binding site, and our substrates need to match that binding site, otherwise they won't fit. And so if we had something come in there that was, as far as the shape goes, if our substrates have, rather than a triangle on this side, if it was two substances that both had kind of a rounded end, and so something like this and something like this, it's not going to fit. It's not going to make it into the binding site. It has to be that specific set. And so you have to have the exact process there. And we'll come across this same idea rather than with enzymes, but with things like neurotransmitters and hormones as well. We have to have the exact right type of binding site for them to work properly as well. When we talk about enzymes, they can work in both catabolic as well as anabolic reactions. And so whether we want to break something down or whether we want to build something up, enzymes can help in those processes that we have. And so when we do things like eat, we have things like sugars. And if we have sucrose or sugar there, and we enter it or combine it with an enzyme called sucrase in this instance, oftentimes enzymes themselves end in ASE. So if it's an enzyme, it's generally going to have an ASE ending to the name itself. That's there. And so our sucrase here comes in and catalyzes the reaction to break down sucrose into glucose and fructose. In the end, to look at this one over here, the enzyme that we start with is essentially the enzyme that we end with. And so it goes in and our sucrose binds to the binding site. And as it binds to that binding site, the enzyme catalyzes the reaction and we end up with our glucose and fructose free by themselves, and our enzyme there is free all alone, unchanged. It's in the same state that it was when it went in. And same thing goes for anabolic types of reactions. And so enzymes can catalyze these just as well. And so now we're going to build something up. And so in this particular case, we can, if we have an excess amount of glucose in the bloodstream and in the body, we may want to store it away for a later time period. And so we can take multiple glucose molecules and combine them with an enzyme. And as they combine with that enzyme, they're going to then be combined together to give us our glycogen molecule. So we're going to have a polymer here with many glucose molecules bound together, branching off, changing and everything. And our enzyme, our glycogen synthase, has gone through the process. It comes out the other end and it can be used again. So it can go back over, find more glucose, and allow it to be entered into the system itself one more time. The enzymes themselves work quite well. As long as, just aware as with enzymes, we want our body to have the ability to work at its normal operating temperature, enzymes have to be at their normal environment as well. And so both temperature and pH can have an impact on how an enzyme functions. And they can both get them to the point where they're not going to function at all. And so this is what we start to see in our lab at this time, is that temperature and pH can potentially change our system. And so if the temperature is too high or too low, our enzyme activity is going to change as well. And so lower temperatures equals typically a slowed down enzyme activity. If we raise the temperature, we're going to get a little bit faster activity. And so the enzyme is going to function a little bit faster as we raise up those temperatures. But if we raise it too high, now our enzyme, which again is a protein, can start to become denatured. And so it starts to change that protein and it no longer functions the way that it's supposed to. With pH, similar situation. So all enzymes have an optimal pH that they will work in. And if it's not at that optimal pH, it's either going to speed up or slow down. And so depending upon what that pH is, and so it could be, say, 7.2 as far as the pH goes. And if we put it into a situation where, say we're eating something and it likes to have a 7.2, and then all of a sudden it goes down into the stomach and it's got a pH down around 3 because we're eating things and the pH has gone up a little bit inside there. Now it's not going to work. its enzyme activity is going to slow way down. And so it's not potentially going to work. And again, if that enzyme is thrust into a situation where it has an extremely acidic or basic solution that it's in, it very well may not work at all. And so once again, that enzyme becomes denatured. And so enzymes themselves like to have just the right situation. They want to be in just the right environment, both in terms of temperature and pH, in order to function at their optimum. And if we take them out of that optimal environment, then they may either slow down too far to where it's not even really working, or even at the other end, we may speed it up too much, and all of the substrate is used up before we have the ability to do what it is that we wanted to. And so next time we'll start to take a look at the metabolism within the body itself as far as our overall processes go.